In this short lecture, we will talk about blastula, and we will begin with general characteristics of blastula. Then we will identify early and late blastula stages. Then we will move on to classification of blastula, and finally we will describe process of implantation. So, what is blastula? Blastula is a stage of embryonic development which occurs after cleavage and before gastrulation. Okay, it is a stage where cleavage divisions have taken place and germ-layer differentiation is yet to take place. It is characterized by the presence of a hollow cavity. This cavity may be fluid filled and this cavity is known as blastocele, and there is establishment of cellular polarity and differentiation. For example, in case of mammalian blastula, there is outer trophoectoderm and inner cell mass. So inner cell mass will give rise to potentially whole body, whereas trophoectoderm will give rise to supportive tissue like placenta. What are the general characteristics of blastula? The first point is already clear, that it is the resultant structure of cleavage. And if we talk about cellular organization, the hollow cavity, which is blastocele, can either be present or absent. If it is absent, we call it solid mass blastula, because in case of solid mass blastula, there is only solid mass of cells, and the cavity blastocele is either absent or very much reduced. And if it is present, it may be present with yolky mass, or yolky mass may be entirely absent. That's how we classify different types of blastula. We will talk about this in a moment. Onset of differentiation. So the cells start to acquire specific fates. I talked about this in previous video as well, where I talked about morula to blastula transition, and I specifically described how zygotic genome activation takes place and how different cells acquire these specific fates. The size of embryo will remain relatively the same. This we also described in the video of cleavage. For the fifth point, there is establishment of polarity. There is establishment of animal vegetal polarity in case of amphibians, and there is establishment of specific inner cell mass and trophoectoderm fates in case of mammals. Blastula marks the stage just before morphogenetic cell movements takes place, and there will be formation of three different germ layers: ectoderm, mesoderm and endoderm, which will mark the stage of gastrula. So it is but blastula that develops and matures into gastrula. How do we identify the blastula stage? In most of the cases, we can identify blastula stage by presence of blastocele, because the solid mass blastula is not that common. Okay, in most of the cases there will be blastocele cavity. If we talk about number of cells, there are typically 128 in many species, but this number may vary with the species. The epithelium will start expressing certain cell adhesion molecule, and there will be tightening of these epithelial layers. There will be compaction stages. We talked about this in the morula blastula transition lecture. There will be functional shift. There will be zygotic genome activation. Different cells will acquire different fates. Okay. The cleavage divisions will begin to slow down and cell cycle will begin to lengthen, or the cell cycle will begin to restore its original duration. How do we differentiate early blastula from late blastula? What are the general features of early blastula? So, early blastula will be formed just after the cleavage. The blastocele cavity may either not be completely formed, or there may be small cavities visible which may be aggregating to form a larger blastocele cavity. The cells are still relatively totipotent, but they will be losing this totipotency, and some of the cells will acquire pluripotent fate and others acquire specific multipotent fate for the supportive tissue. There will be minimal morphological differentiation, means most of the blastomeres look exactly the same, but some blastomeres may be somewhat different due to differential gene expression. The zygotic genome activation may still have started already, and the cell cycle stages will start to slow down. The cell cycle will start to restore to its original timings. In case of late blastula, we will see a well-developed blastocele, which is a large cavity. The lineage segregation is more evident. In case of amphibians, there will be clear animal vegetal polarity, and in case of mammals, there will be clear differentiation of the outer trophoectoderm. As you can see here, this layer is trophoectoderm, and we have the inner cell mass. But in this diagram, this very much outer layer is the zona pellucida, which is still intact. The cell cycles slow down, G1 and G2 phases will start appearing and lengthen up, and the embryo will be primed for gastrulation movement. As soon as the embryo is implanted, the gastrulation processes will begin and there will be formation of 3 specific germ layers. Now we will study the classification of blastula. The blastula is classified on 3 different bases. The first one is amount of yolk present in the egg from which blastula is forming. Second, the distribution of yolk in that egg, whether yolk is distributed uniformly, whether it lies at vegetal axis, vegetal pole, whether it is concentrated or whether it is lying as yolky mass in the center. And third basis of classification is the cleavage pattern. What kind of cleavage pattern has been observed, whether it is holoblastic cleavage, whether it is meroblastic cleavage, discoidal superficial. The cleavage pattern also influences what type of blastula will be forming in that specific case. Coeloblastula, and this coeloblastula is formed from microlacithal eggs or isolacithal eggs. The eggs where yolk amount is either very less and yolk is uniformly distributed. The cleavage type is holoblastic cleavage, which is specifically holoblastic equal cleavage. To know more about these terms, please watch the cleavage video. And the examples are echinoderms, in which we study sea urchin, and amphioxus. Talking about the structure of coeloblastula, it is a spherical blastula with large hollow cavity, we call blastocele, and blastomeres are present only at the periphery. The only thing we have to remember in case of coeloblastula is that it is formed from microlacithal eggs, and these microlacithal eggs will have uniform distribution, which is isolacithal conditions. The cleavage is holoblastic. The blastomeres are arranged on the periphery most of the time in single layer, and there will be a large hollow blastocele cavity. The examples are echinoderms. Let's move on to discoblastula. The discoblastula is formed from the eggs which have huge amount of yolk, the macrolacithal eggs, and yolk may be most of the time distributed to only one axis or only one position. For example, the yolk is distributed to this basal half. So such eggs are telolacithal eggs. So we say the macrotelolacithal eggs will form a blastula we call discoblastula. And the reason is simple. Most of the egg is covered with yolk and there is very less cytoplasm at the periphery of one axis. Let's say this is animal axis. So when the divisions, the cleavage takes place, the cleavage is meroblastic discoidal cleavage, will result in smaller cavity. So at animal pole you will see a blastocele cavity and the periphery is arranged by all these blastomeres. The blastocele is relatively flattened and smaller in size. It appears like a subgerminal cavity of disc shape. The most common examples are fishes, reptiles and birds. Let's come to one of the most studied blastula, which is amphiblastula, which is seen in case of amphibians like frogs. So it is formed from mesolacithal eggs, means moderate amount of yolk, and yolk is mostly distributed to one end or one axis. Like here we can say that yolk is distributed to vegetal half of the egg. So there is mesolacithal eggs and cleavage is holoblastic complete cleavage but unequal, because this yolk will hinder the cleavage process itself. This will result in two different kinds of blastomeres, because the first cleavage has not completed and second cleavage will begin at animal poo. This will result in the cells or the blastomeres of animal half being much smaller. We call them micromeres, and the yolk is restricting the cleavage, so there will be large sized cells in case of vegetal half. So we have two types of cells. We have micromeres at animal half, we have macromeres at vegetal half, and this difference in size is due to this holoblastic unequal cleavage, which means that only, for example, only first cleavage is taking place around this vegetal half, and second or third cleavage may already be started at animal half. Now let's come to periblastula. It is a specific case of insects. It results from central acythal eggs. Like you can see that there is central yolky mass, and the cleavage is meroblastic superficial cleavage, which is characteristic feature of many insects. The most studied insect egg and blastula is of course Drosophila. If we talk about structure, there are peripheral blastomeres only. There is central yolk mass which is not cleaved. And finally, we will talk about the human blastula, which is termed as blastocyst. Now, this blastocyst is a modified coeloblastula, and it is modified in such a way that the outer layer, zona pellucida, is not lost yet, and the inner layers are differentiated into two specific layers. We get outer trophoectoderm and we get inner layers of inner cell mass, and opposite to this inner cell mass lies a cavity which is called blastocele cavity. So from what type of eggs do this blastocyst will form? It is mostly formed from either microlacithal eggs or alacithal eggs. The cleavage is holoblastic, which is complete cleavage, and it is modified for baby parity. That is why there is either very less amount of yolk, or yolk is entirely or practically absent. We have a comparative table of different types of blastula, their structure, the basis of their formation, which is the type of egg, and type of cleavage which results in formation of such blastula, and specific examples. Now we will move on to maturation and implantation, and I will refer blastula as blastocyst because in case of humans the blastula is termed as blastocyst. So first there will be formation of blastocyst, which is around day 5 or day 6 after the fertilization itself, and the first process in this maturation is the blastocyst expansion, which occurs around day 6 to day 7 after fertilization. In case of expansion, the trophoectoderm cells will start pushing fluid inside, which will result in the enlargement of this blastocele cavity. The blastocele cavity expands in size, and zona pellucida will thin out due to exertion of pressure from within or from inside. The trophoectoderm will also form this tight epithelium due to expression of the tight junction formed by these cell adhesion molecules. If you want to know about the details of all these processes, I have talked about these processes in the video of morula to blastula transition. The inner cell mass will start differentiating into epiblast as well as hypoblast. The epiblast will be forming the embryo proper, and the hypoblast, which is primitive endoderm, which will later form endoderm, will be forming some extra embryonic supportive tissue. It will contribute in yolk sac as well. We have formation of blastocyst and we have expansion of blastocyst, which only have two purpose. First is expansion of blastocele cavity, and this expansion will exert pressure on zona pellucida so that zona pellucida will thin out, because there is a specific need of zona pellucida to rupture. It is zona pellucida which is preventing the contact of this blastocyst with uterine cavity. Once this zona pellucida is damaged or ruptured, the implantation process can finally be taking place. There is blastocyst expansion, and it is followed by hatching from zona pellucida, which occurs around day 6.5 to day 7, and it occurs just before implantation. Okay, so zona pellucida will break down due to this heavy exertion of pressure, the blastocyst expansion and contraction will exert the mechanical pressure, which will result in breakdown of this zona pellucida. The second thing which contributes to breakage of this zona pellucida is proteolytic enzymes, and these proteolytic enzymes are secreted by the cells of trophoectoderm itself, and they include trypsin like proteases, and the maternal uterine environment will also contribute to this hatching process, because the uterine secretions will also contribute some of the proteases, and it provides the suitable habitat, the suitable acidic pH for all this process to happen. And finally, this blastocyst hatches and becomes free in uterine lumen. So, what is the significance of this process? What is the significance of this hatching from zona pellucida? As we talked about earlier, it is what zona pellucida which prevents the interaction with the uterine cavity. So, it is preventing ectopic implantation in oviduct. It is timed in such a way that this zona pellucida will just break when the blastocyst is at proper positioning, proper uterine position to implant itself. And this hatching is essential for physical contact with endometrium of uterus. Next process is apposition, and this apposition will signify the initial contact with uterine epithelium. The blastocyst will come in close contact with uterine epithelium, and it is the cells of inner cell mass. This portion will make the contact, as you can see in this figure. For this apposition to happen, the uterine endometrium must be receptive in its nature, which means that the uterine epithelium will be under the influence of hormones like estrogen as well as progesterone, and there will be a specific implantation window when this hormonal regulation takes place, which is around day 20 to 24 of the menstrual cycle. The extracellular matrix of this uterine epithelium will start secreting the complex sugars. There will be collagen, laminin, fibronectin, specific cadherin, which are a cell adhesion molecule. We have hyaluronic acid, and we have some specific heparin sulfate receptors. All these molecules will help in the implantation, because at the receptive phase, this uterine endometrium has prepared itself for the implantation. So, let's talk about the adhesion phase. That what are the molecules, what are the specific molecules involved in adhesion of this specific blastocyst, but leads to implantation of blastocysts into the uterine endometrium. As you can see, we have two cells. Here we have the cell of trophoectoderm. Here we have the cell of uterine endometrium. First of all, let's look at the polarity of the cells, that but phase of cells faces but phase of other cell. The basal surface of trophoectoderm will face the apical surface of uterine endometrial cell. This is the first point. And the apex of the trophoectoderm will face toward inner cell mass as well as blastocele cavity. Okay, so there is expression of certain cell adhesion molecules which is signified by this outbranching here by both the cell of trophoectoderm as well as the cell of uterine endometrium. And as we already talked about, in the receptive phase, there is a specific molecular secretion like fibronectin, laminin, collagen, and here collagen is specifically type 1 as well as type 4, and certain type of proteoglycans. In case of the specific trophoectoderm, we have secretion of similar molecules as well, some of which are cell adhesion molecule, and some of the molecules will provide other specific interactions as well. In case of endometrial cell, we have similar cell adhesion molecules, and some of the molecules will act as some receptive kinds of molecules. These integrins will bind to extracellular matrix. I will be referring ECM to extracellular matrix from now on. The integrins will bind to this fibronectin, laminin, collagens. The P cadherins will bind specifically with P cadherins of receptive endometrial cells. The HSPG, which is heparin sulfate proteoglycans, these will also bind to ECM molecules. The L-selectins will bind to L-selectin ligands, which will be expressed by these endometrial cells. The MMPs will interact with all these ECM molecules. The syndicands as well as CD44 will bind with some proteoglycans present in extracellular matrix as well. And these MMPs will also interact with the hyaluronic acid receptors and some other receptive proteoglycans as well. So these are the specific interactions which will be leading to making in contact and finally in the reception of this blastocyst and the process of implantation at last. So this table comparatively determines that these are the trophoblast cell adhesion molecules like integrins bind to fibronectins, laminins, collagens, HSPZ, and here is a specific mechanism like the basal integrin binds to ECM secreted on the apical surface of uterine epithelium. We saw that uterine cells faces apically, whereas this trophoblast cell faces basally, and there will be adhesion and signaling which will finally be resulting in implantation as well. The peak adherin will be binding to uterine epithelium through peak adherin or luminal peak adherin on luminal epithelial cells, and it will result into homophilic binding because the peak adherin on trophoblast as well as this uterine epithelium will be of the same nature. This is specific cadherin interaction, and cadherin is calcium dependent, and there is docking of trophoblast to uterine epithelium due to this p cadherin interaction. There is heparin sulfate proteoglycan which will bind to this ECM molecules like collagen, fibronectin, and laminin, and there is a very small or very minor interaction with uterine epithelium or uterine cell adhesion molecules, and it basically strengthens the integrin interaction. The L-selectins will bind to glycoprotein ligands which are present on uterine epithelium itself, and it mediates the initial rolling or tethering of this blastocyst. In case of MMPs, which are matrix metalloproteinases, it helps in this ECM remodeling, which is essential that we degrade some of the ECM to allow invasion, because this uterine epithelium has secreted this specific ECM, and these extracellular matrix will have to be modified. Some of the molecules will have to be degraded to make path for the blastocyst to implant itself. So it facilitates the adhesion indirectly. For syndicans and CD44, they will interact with ECM proteoglycans like heparin sulfate and hyaluronic acid, and they will act as co-receptors. They will help integrin binding as well as some specific signal transduction, which will finally help in completion of all these processes of invasion as well as implantation. Now, final stage, the stage of invasion. In case of invasion, the trophoectoderm will differentiate into, as you can see, syncytial trophoblast, which is outermost layer, and it is invading directly into these endometrium, and we have cytotrophoblast, which is the inner layer. This cytotrophoblast is mitotically very active, and the process of gastrulation is about to begin very shortly. These matrix metalloproteinases will help the syncytial trophoblast in this invasion process itself. There is establishment of contact with maternal blood vessels, and it is for the very first time the beginning of maternal fetal interface. What is the endometrial response? The stromal cells of endometrium will undergo decidual reaction. There will be accumulation of glycogen, some specific lipids, and some specific cytokines like vascular epithelial growth factors, and there will be establishment of immune tolerance. For implantation, what is the initiator? The contact with uterine endometrium, this contact which has already been established. There is something which is known as vent signaling pathway. This vent signaling pathway will remodel the uterine ECM as well as it allow the blastocyst to embed deeply within the uterine stroma. Okay, so wind signaling will help in ECM digestion, remodeling, and ingestion or invasion, complete invasion of this blastocyst into uterine stroma. The trophoblast will help in this process by releasing certain proteases. It will release the collagenase which will break down collagen fibers. It will also secrete stromycin, which will digest proteoglycans and glycoproteins of extracellular matrix, and it will also release the plasminogen activator, which will convert the plasminogen into plasmin, and this plasmin degrades the fibrin and extracellular matrix proteins. In short, all these enzymes will make a pathway for the implantation for this. They will make a pathway for this blastocyst to invade the uterine stroma as well. You can see this figure A shows a mature mouse blastocyst. The zona pellucida is still present. There is outer trophoectoderm, there is inner cell mass, and there is large blastocele cavity, which determines that this is late blastula stage. And due to the internal pressure, some enzymatic activity, hormonal action, there will be breakage of this zona pellucida, and this blastocyst will escape this zona pellucida layer. The blastocyst will uterine cavity. The uterine cavity is perfectly receptive, and there will be implantation taking place, as you can see. And the invasion is taking place from the side of inner cell mass. There is outer trophoblast as well.